It means that somehow or another we are not communicating. Some of the class members are getting it. They know the answers, they get them down. Others simply don't. So the question is why? And what can we do about that? I'd like everybody to score a high grade, not necessarily for the grade. I want you to learn something about biology in this class. And if you're getting low scores, it indicates that you're not learning much, if anything. So you may have noticed that one of the assignments I sent out was for you to submit to me questions about things that that you wonder about, or you don't know the answers to, or you're confused about. And then we'll discuss them next time. We'll discuss the questions that you have and see if that helps you to clarify and understand the material, so you'll score better on tests. On that note, the next quiz, quiz 6, will be on Monday the 22nd, and test, so that will be just a quiz, and test 2 will be on Wednesday, March 24th. Now, I didn't really want it on that date, but it turns out that the next class period is the 29th, and the 29th is in spring break. And I don't want to wait till after spring break to immediately give you a test. So we'll have a quiz on Monday and test 2 on Wednesday. And test 2 will cover everything that we have that has not been tested since test 1. And I indicated what those topics are in the announcement that I sent you. Any questions about that?
Professor, I just have a question. So last week I resent the worksheet into the campus, and I wonder if you see that or not, because you told me that I need to resubmit the worksheet that due last week.
Yes, I think I got that and graded it. Have you looked at your?
No, my grades have not changed yet. It's still the same.
Oh, okay. Then that means I haven't.
Okay.
Okay, I will check on that.
Okay, thank you.
Anything else?
Are we going to be doing the pairings this week, or?
No, that will be done on April 7th.
All right.
All right. Well, if nothing else, I'll go to the quiz from last week. Okay, can you see the quiz 5? Is it large enough for you to be able to read?
Yes.
Yes.
Okay. All right, I'm going to have you volunteer to answer these questions. Basic structure of DNA polymer is commonly described as a what stranded double what?
I'll go Taylor. It is a double stranded double helix.
That's right. And that question is taken directly from the slide that's shown there, showing a double-stranded double helix. The next question is about the discovery of the DNA molecule and who got the Nobel Prize for it. Watson, Crick, and Rosalind Franklin were all involved in the discovery. The question is, were they the ones that got the Nobel Prize in 1962? And the answer for that is what?
I'll go. My name's Abby Beck. That was false.
And why is it false?
Sorry, my dog's walking in the background. I think it has something to do with the woman, Rosalind Franklin. I don't remember off the top of my head.
Yeah, you're right. Rosalind Franklin provided the key picture, X-ray picture of a DNA molecule that tipped off Watson to how to construct models to arrive at the structure of DNA. Well, it turns out that that was in the 1950s and women weren't necessarily given all the credit they deserved. And then to complicate things further, Rosalind Franklin died in 1958, and this award was not until 1962. So she was not included in the Nobel Prize. All right, next, fill in the blank. We studied several types of RNA, mRNA, tRNA, rRNA, and what?
Ribosomes.
Sierra, okay.
Yeah, that was Sierra.
Ribo what?
Ribosomes.
Ribosomes?
Am I saying it wrong?
Well, to me, it sounds like you're saying ribosomes. And are you saying ribozymes?
Yes. Hmm.
Okay.
Sorry.
Well, the acoustics aren't great here. Yeah, it's ribozymes. And this slide shows where that comes from. Ribozymes are a form of RNA that is catalytic. It's the only form that is catalytic. Okay, the next one. The CFTR protein has two nucleotide binding domains. What is the function of these regions with respect to the protein's function, which is to transport chloride ions? And the slide from which this question came is shown there. So who wants to give the answer? Anyone? I gave credit to a broad array of different types of answers, but they all had to include certain things. So what's the essential feature?
It's for replication. There's, oh, sorry.
Robert Johnson, you were
Professor?
okay.
Yeah, the nucleotide binding energy generates energy to facilitate transmembrane transport of chloride ions.
Yeah. So you said the correct words, providing energy to transport the chloride ions across the membrane. So the hydrolysis of the terminal phosphate on adenosine provides the energy to get chloride ions across the membrane. And you end up with ADP after that happens. Okay. Next, terms leading strand and lagging strand refer to what process with respect to DNA? Someone
The replication.
who said that?
Rosemary.
Okay. Replication, that's right. Not duplication. It actually is a duplication, but the term that's used for copying DNA is replication. And then number 6, a mutation is defined as any change in the amino acid sequence, true or false.
False.
Who said that?
Hilberg.
Okay, Hilberg. And you said it was what?
False.
Okay, and why is it false? It says so on the slide there.
Can't really read that.
Okay, well, you should know what it says. Let me make it a little bit bigger. Wherever you see the word definition, memorize that. In this case, the definition is a mutation is a change in the nucleotide sequence of DNA, not the amino acid sequence. So the mutation is always at the level of DNA. The consequence of that is a change in the amino acid sequence. Could be, not necessarily. Remember, there are silent mutations. So a mutation can lead to a change in the amino acid sequence, but not necessarily. The next one, multiple choice. Which of the following terms is not associated with Okazaki fragments?
Kiyomi
Continue.
Anderson.
Okay,
Sorry. Continuous.
yeah, go ahead.
Continuous.
Yes, continuous is right. All of the other terms apply to the lagging strand, which is where Okazaki fragments are formed. Continuous, they're not formed because going from the 3' to 5' direction on the leading strand never stops. You never have to stop and restart again. Okay, so that's where that one came from. Then a short essay here. Exposure of DNA to UV radiation can cause a thymine dimer, which produces a kink. What effect does this kink have on protein synthesis?
I put this is Justin Hansen. I put it would cause it to mutate.
And that's right. Not necessarily, but is a very good chance that when DNA polymerase goes copying along on that strand, when it comes to a kink, it will make a mistake. And that would be a mutation. So that's the answer. And this is a slide that the question came from. Next, multiple choice. Transfer RNA molecules function to shuttle individual amino acids to the ribosome during translation. How many different types of tRNA molecules would a human need to make all the required proteins? You have 4
Katrina Vibbard. It would be 20.
That's right. 20. That's the number of essential amino acids that there are, and each one has its own unique tRNA, which is quite remarkable. And finally, the last one. Not many people got this right, which surprised me because we spent a lot of time talking about DNA sequences and mutations. So who wants to answer this question?
Kiyomi Anderson.
Okay,
It's deletion because GGA is missing.
yeah, that's correct. And why is it not, for example, a frame shift? How do you know that? Anyone?
If it was a frame shift, it wouldn't be less, but it would just be one. Each one would be one off, rather
That's
than
right.
this is just
That's right.
completely.
So
Yeah.
if it was a frame shift, everything from the point of the mutation downstream would be different. And in this case, it's all identical. Only thing that's missing is that one codon, GGA. So you can eliminate all the other kinds of possibilities and come to the conclusion it has to be a deletion. All right, anything else on this quiz? Any questions about any of the questions or the answers to them?
Professor, I don't know why, but I feel like the quiz is like kind of too rush for like answer all like the 10 question in 15 minutes. I don't know, it was just me or I don't know, because like some of the question I have not like have enough time to actually like writing it down and choose the questions.
So I have that problem too. So basically, like for more time, if you give us more time, it will be way better.
All right, I sent an announcement about my policy on time constraints earlier. I'm not going to change that. I can, and I don't want to, for example, for a quiz, I don't want to make the time limit longer. And the reason for that is it eats into class time. And if we keep making the length of the quiz longer, we have less and less time to do classwork. So if there is a problem, the answer would be two things. One is I can make I can put fewer questions in. And two, you can study well in advance. So you know the material and you don't have to spend a lot of time thinking about it. You see a question and you immediately know the answer to it. So I will take that into consideration. Reduce the number of questions. You try to get yourself well prepared in advance.
Professor, I don't think it's the number of questions. I think what it is there's multiple parts to a question. So like for me, when I take the quiz, I go through and I try to answer what I can first and then do the essays at the end. And then that way, if I run out of time, at least I got down what I know. But sometimes some of the questions have multiple parts. So you spend a little longer on those questions. I don't think it's necessarily the issue that there's 10 questions. Just sometimes some of the questions are 2 or 3 questions within themselves.
Okay, let's take quiz 5. It's up here on the screen. As an example, what question or questions would fit the problem that you're describing? I'll just page through it, see if you can spot one that gave you a problem.
I would say probably number 4s and number 10s.
Number 4, the nucleotide binding domains.
Yes, and number 10s. Because number 10s, at first I was like kind of look at it and I thought it was like a silent one, and then like I look back and then I thought it was the misstance, but I ran out of times, so it was just keep like the wrong answer.
Okay, well, I guess what I'm searching for is how would I ask that question in a different way? You're saying that it's too complicated, but I want you to let me know that you know how to read a DNA sequence and that you know what the types of mutations are. So I'm struggling with how to change the question to be less confusing to you.
Basically, I don't know who else like thinking this way, but for me, I have a really bit problem with reading. I'm not like, I can't like read fast. I have to like slowly read the question, and that's might take my long time. I don't know like if somebody else like that, but for me, because this is my third language, I don't know, it's a little bit hard for me.
Okay. You should see DSPS. If reading is a problem, you can have, get DSPS approval for a change in the time allowed. A number of students have done that, and I've accommodated them in every case.
Okay, thank you.
All right, well, I'm appreciating your feedback. I'm just trying to get my teeth into what I can do on my part. I've said what I think you can do. But in terms of constructing a quiz, you've said that changing the number of questions isn't important. 10 questions is okay.
I think there's too many questions for 15 minutes, but that's, I guess some people don't think that, but
You think there's too many? Well, all right, for quiz 6, next Monday, I'll go for fewer questions and see if that changes the scores. We have to do something, and it's not the scores, as I said before, it's not the scores that bother me so much, is that the scores reflect that you're not really learning the material. And that's the goal of the course. If you're not learning, there's something wrong, and you're wasting your time spending time in the course and not learning anything, or learning very little. So I will shorten the quiz. I will try to make the questions clear, but as you can see, I take the questions directly off of slides in the lecture. So if you're studying those slides, and if you're spending 4 hours a week outside of class time studying the material presented in class, you should have the answer to every question. If you remember reading the syllabus, the recommendation is that you spend 4 to 6 hours outside of class studying the material for this course in order to do well.
Professor, this is Rosemary. Would you be able to go down to that other, I think, question 3 again? I believe.
Yeah.
Or, sorry, one more. Question 4.
Question 4, okay.
Okay. Would you be able to explain this just one more time?
So I said, what is the function of the nucleotide binding domains in the protein's function? So the protein we're talking about here is CFTR, cystic fibrosis translocation. Can't remember what R stands for, regulator. So that protein has to transport chloride ions.
Mm-hmm.
And what is the function of the nucleotide binding domains in the transport of chloride ions through the CFTR? And the answer is it provides energy. The nucleotide binding domains provide energy. And they do so by hydrolyzing adenosine triphosphate, ATP, hydrolyze off the terminal phosphate, and that releases energy to change the structure of the protein channel so that a chloride ion can be transported across. Now, you didn't have to say all that in your answer. If you said anything at all about production of energy to transport, I gave you full credit for it.
I think when we were in class, you had said it changes like the shape of the canal, I believe.
Yeah.
Okay. I just wanted to make sure. Okay. Thank you.
That's what the energy does. It changes the shape of the protein so that the chloride ion can pass through. But the key word here is energy production.
Okay. Thank you.
Okay, anything else? I want to reiterate that I appreciate the feedback that you're giving me. Let's hope it results in better outcomes. All right, if nothing else, I'm going to stop sharing that screen. Now you should see membranes and transport. Yes?
Yes.
Okay. What is a membrane? A selective barrier that allows some things to go through and others not. Things like molecules, ions, and small particles. What is transport? It's movement of an object from point A to point B. What is a plasma membrane? It's primarily a phospholipid bilayer, but it also contains proteins, among other things, and it forms a barrier between the cell's contents and the exterior of a cell. And on the right here, you see a transmission electron micrograph showing two plasma membranes, two cells, cell 1 and cell 2. You can see the railroad track outlines of the two plasma membranes with the intercellular space between them. And you can actually even see the lipid bilayers in certain regions, the little space between the two. What are the functions? Well, they're semi-permeable, which means that they let some things pass and not others. The membrane controls exchange of materials, communication, cell-cell recognition, transformation of energy, and cell adhesion. Well, what are the components that make up the plasma membrane? Well, there are four major classes of biological macromolecules. The plasma membrane contains three of them. And what the plasma membrane contains is listed here: lipids, proteins, and carbohydrates. Which one is missing? Of the four polymers that we're working with, that we have studied, which one is not found in plasma membranes? Anyone?
Wouldn't it be nucleotides
Nucleic acids, yes.
or yeah.
Right, so nucleic acids are not found in plasma membranes, but all the other three macromolecules are. Among the lipids are the phospholipids, of course, but then glycolipids and glycoproteins, and steroids, in particular cholesterol, which we'll come back to later. Cholesterol is very important. The other thing is that I want to mention here is that there is on the plasma membrane a glycocalyx, which is also called an extracellular matrix, and it's composed of proteins and carbohydrates. We'll talk about that next. What is the glycocalyx? It's a thick, ultra-sticky, gel-like covering of the plasma membrane. It's composed of sugar and protein strands bound together. On the right, you see this hairy material on the surface of the cell, on the endothelium. That's the glycocalyx. And pictorially, it looks like this, these long chains of amino acids and sugars that are all twisted up together to make a thick, stewy mass. And you can see a cross-section of a bacterial cell here looks like it has a hairy surface. That is the extracellular matrix. So what is it? What is the function of this structure? Well, for bacteria, adhesion, in fact, for all cell types, it's involved in adhesion. Cell hydration to avoid drying. For those of you who fish, you pick up a fish, it slips out of your hand. It's because it has a glycocalyx on its scales. Modulation of red blood cell volumes, and a shield protects bacteria that are trying to attack. It protects them from the immune system. This is to emphasize the equivalence between the term glycocalyx and the term extracellular matrix. For animal cells in particular, this matrix is important as a structural feature. Animal cells do not have cell walls, but they need structure, and the glycoproteins provide that in animal cells. So they're not as rigid as plant cells, but they do have some degree of rigidity. So let's take a closer look at the phospholipid bilayer, which is the main component of a membrane. It's constructed from phospholipids. There are two layers, and they separate the outside of the cell from the inside. The outside of the layer is hydrophilic. That's because it's facing water. The inside of the layer is hydrophobic. You see that spelled out in this picture. The exterior is watery, the interior is watery, but the membrane accommodates both the watery surfaces, and inside it's hydrophobic, and it has to be hydrophobic in order to provide a barrier. Otherwise, if it weren't for the hydrophobicity, the thing would just dissolve. And you remember the structure of a phospholipid molecule. We've talked about this before. It's a hydrocarbon chain, usually a long chain, with a phosphate group on one end and another molecule, a polar molecule attached to the phosphate. This hydrocarbon chain can be either saturated or not. You see that up here in the upper right. If the carbon chain is saturated, it's long and straight and rigid. If there's at least one double bond, the chain can bend, and so it's flexible, and that makes a big difference in the structure of the membrane. I want to you've had this before, I want to mention the term amphipathic, because phospholipids are amphipathic molecules. Well, what does amphipathic mean? It means that on the one end of the molecule is hydrophobic, the other end is hydrophilic. And that is critical for the structure of the plasma membrane. Now, where else have we run into that term? If you remember when we were discussing digestion, discussing the pancreas, and the gallbladder produces bile, which contains bile acids, and bile acids are amphipathic. Here's an example of one, glycocholic acid. It's a steroid on one end, which is very nonpolar. On the other end, there's an amino acid, which is polar. So the amino acid is compatible with water. The steroid is compatible with fats and oils. So what does this acid do? It has two roles. When you eat fat, it emulsifies the fat and solubilizes the emulsions so that enzymes in an aqueous environment can act on the fats, the fat breakdown products. So these are two different, completely different cellular functions, both involving amphipathic molecules, digestion of food and structure of membranes. So another example of amphipathic molecules are soaps. Soaps are surfactants. And any soap molecule has a hydrophilic head and a hydrophobic tail. And the way they deal with nonpolar substances like oil is that the nonpolar tails go into the oil and they make a sphere, the polar head sticking out into water, and this is called a micelle. So I thought I would digress just a bit and talk about soaps, because soaps are really, they're very much a part of our lives, and they're also very simple. A prototypical soap was historically made from ashes and lard. So the chemistry of soap is interesting. The way it was done, and you can see some pictures, some old photographs of people making soap here, boiling ashes in a big kettle. And on an industrial level, these big vats were filled with ashes and water and boiled. If you want to do it in your kitchen, you can suspend ashes in water, boil for 30 minutes, and then add cooking fat or lard, and then add more lye water until it gets thick. And then you see here pouring it into a mold. And after it hardens, it can be cut up into bars and used as soap. So what is the chemistry of this? In the blue section here, the basic ingredients of a triglyceride. Remember that fats are triglycerides, which are glycerol with long chain fatty acids attached to them. So if you take a fat, mix it with ashes, what do the ashes provide? Sodium hydroxide. What you get is glycerol is released and replaced with sodium ions, and that is a soap. In this case, the soap is sodium laurate, which is has a long nonpolar tail and a polar head, which is a sodium ion. So how does, when you wash your hands, how does a soap get the dirt off? Well, that's illustrated here on the lower right. You have the soap molecules, which are amphipathic, polar and nonpolar. You have dirt, which tends to be nonpolar. So the nonpolar tails attached to the dirt particle surround it. So with the polar heads in the water that you're using, and that washes the dirt away. So if this is skin, the polar groups that are soluble in water lift the dirt away from the skin. Any questions or comments about amphipathic in particular, or soaps? Then we'll go on to the steroids and glycolipids. Cholesterol, this is the structure of cholesterol, is inserted in the phospholipid bilayer at various points, and it can either increase or decrease membrane fluidity. And with that, I'd like to introduce the concept of the fluid mosaic membrane, and it's based on a model. The phospholipid bilayers are semi-solid, which means they're fluid. A fluid is something that can flow, and a semi-solid can flow. The membrane is a collage of proteins embedded in this matrix. And it's not homogeneous. There are microdomains present throughout the membrane. And what you see in these pictures, first of all, an electron micrograph, again showing the phospholipid bilayer of a membrane, and then a drawing of the same thing, showing that these proteins are embedded in the phospholipid bilayer. Some of them traverse the entire membrane, others are just on one side of it. We'll talk about those a little later. So the details of the fluid mosaic membrane, this is important. You really need to know to have an idea what a fluid mosaic membrane looks like and how it functions. So the statement of the model, the membrane components, which are the proteins and glycolipids, form a mobile mosaic of islands floating in this sea of phospholipids. At room temperature, the plasma membrane has a consistency of vegetable oil, so it's semi-solid. And this allows the components of the membrane to float. So the membrane isn't static, but it rather is in constant motion. Now, what are the factors that affect fluidity? And the fluidity has to be just right in order for the membrane to function. And remember what its functions are. For one of the main ones is transport of materials from one side to the other. So the fluidity has to be at just the right level in order for that to occur. As the fluidity increases, permeability increases up to a point. If fluidity gets too high, the membrane falls apart. If it's too low, it becomes non-functional. So what are the factors that affect the fluidity? Well, temperature is one. The fatty acid degree of unsaturation is another. And the cholesterol content is a third. So remember those three things: temperature, fatty acid saturation, and cholesterol. This picture on the right shows, first of all, a saturated fatty acid where the nonpolar tails are straight, rigid, stiff, whereas one that has an unsaturation, the tail is bent, causing space between the tails. So a membrane that has unsaturations will be more fluid at lower temperatures. And you can see the temperature curve here. The membrane is solid-like at low temperature. There's an inflection point, and above that, it's more fluid-like. Now, what does cholesterol do? It inserts between the nonpolar chains, and it acts like a buffer. At low temperature, cholesterol increases fluidity, whereas temperature tends to decrease fluidity. If the cholesterol content is high, it would prevent that. And at high temperatures, it does the opposite. It decreases fluidity. So it's kind of a buffer. As I said, if fluidity is too high, the membrane falls apart. If it's too low, it can't function. So this slide is to try to help you envision what a functional membrane is actually doing. The membranes in your body are doing this as we speak. So remember that the bilayer is a thick, viscous semi-liquid. The proteins and glycolipids embedded the membrane are in continuous movement, floating around like a raft on a pond. So all of these proteins here, you can just imagine, they're sort of bouncing around while the membrane is in motion. So any given protein does not stay in one spot. It's constantly moving within the membrane. And there are peripheral proteins which are only attached to one side, and I think almost always on the inner surface. Peripheral proteins are attached, and the attachment is reversible. They can come and they can go. And examples of this would be hormones, peptide hormones, or antimicrobial peptides. On the other hand, the proteins that go all the way through the membrane, they're called integral membrane proteins, and they are permanently attached. They can't come and go as they wish. Once they're there, they're locked in. And they're the ones that are involved primarily in transport across the membrane, either from inside to outside or the reverse.
So Professor, I'm confused. What do you mean by that? They use like lily pads, they use that to get in and out of the membrane and those don't move? Or what do you mean by those are used for in and out if they're permanently attached?
They're permanently attached, but they don't stay in one place.
Okay.
So any one of these long blue tubes, we're showing them in a still picture, but in real life, they'd be bobbing around like a raft on a pond in this semi-liquid, semi-solid, I should say, bilayer. The bilayer, as I said earlier, it has the consistency of vegetable oil. So it's like, it's between a solid and a liquid. It's a semi-solid. And things, the permanent proteins, the ones that transport things, are there, but they don't stay in one place. They're constantly moving around. That makes sense?
Yes, thank you.
Okay. We're going to move, hopefully you all have a good understanding of what the fluid mosaic membrane model is. It's this. We're going to move from that now to a consideration of diffusion and osmosis. What is diffusion? Again, here we come with a definition. It's a movement of molecules from higher concentration to lower. You've all done the little exercise you see pictured here to the right. Take a drop of food coloring in a glass of water. And at first, when the drop hits the water, it's all concentrated in one place. But with time, you don't have to do anything, just leave it. And with time, it disperses until finally it's evenly distributed throughout the water. And that's based on the kinetic energy that's intrinsic to molecules. They're constantly moving around and they can disperse themselves. A solute can disperse in a liquid simply because of the inherent kinetic energy that each of the molecules has. So that's diffusion. It's passive. It requires no energy. It just happens in solutions. Osmosis definition, on the other hand, is diffusion, but through a semi-permeable membrane. So remember that. The movement of a solvent, and in biological systems, a solvent is water, through a semi-permeable membrane into a solution of higher solute concentration. So what is a solvent? It's a liquid that can dissolve substances. What is a solute? It's a substance that can be dissolved. So let's look at the picture on the right where we're pouring salt into water. We have two beakers here, and they're connected by a tube. And in that tube is a membrane that allows water to go through, but not salt. So what happens if you look at the level of the water in these two beakers, you see that the one in the right-hand beaker, the water level goes up, corresponding to a lowering of the water level in the left-hand beaker. And the reason for that is the salt. Salt can't go through, and so water migrates to the salt molecules. And if you remember when we talked about what a solution is, it's the hydrogen bonding of water molecules to the ions, in this case, chloride ions and sodium ions. That hydrogen bonding of water molecules is what is responsible for dissolving the salt. And osmosis is this diffusion of the water through this membrane. Then we have to talk about tonicity. Tonicity is a little hard to understand. It's a little hard to apply these terms, but let's go through them, and then you have a worksheet which will allow you to apply the terms, the tonicity terms, to real situations. So an isotonic environment means that the water concentration outside the cell is equal to the water concentration inside the cell. Most cells contain about 0.9% dissolved salts. Interestingly, that's the concentration of salt in seawater. And remember, all life came from the sea. So we still have this characteristic of 0.9% salts in our cells. So an isotonic environment for a cell has to contain 0.9% solutes. And that usually means dissolved salts. So if you want a cell to be happy, you have to bathe it in 0.9% salts. And when you go to a hospital and have an injection, saline solution is 0.9% dissolved salts. Okay, hypotonic. With respect, we refer to these terms with respect either to the inside of the cell or the outside of the cell. Well, hypotonic with respect to the outside of the cell, the environment of the cell, the water concentration outside the cell is higher than inside. Conversely, it means that the solute concentration outside the cell is lower than inside. And the result is that water moves into the cell, causes the cell to swell up. That's hypotonic. The solution with the lowest concentration of solute is described as hypotonic. Hypertonic, and again, this is with respect to the environment of the cell, the water concentration outside is lower than inside. As a result, the water moves out of the cell and the cell shrivels up. The solution with the highest concentration of solute is described as hypertonic. Here you can see it illustrated. I like to use these terms. You may or not find them useful for yourself thinking about this. But for me, hypo means below. And that means that inside the cell, the water concentration is low and solutes are high. So water flows into the cell. It's called cytolysis or deplasmolysis. If the osmotic pressure is high enough, the cell will actually burst from water flowing into it. Hyper means above. So above is high. The cell water is high. Solutes in the cell are low. Water flows out of the cell, and you have what's called plasmolysis. Of course, iso means equal. It's the same. Water is the same both inside and outside. So you can see in the case of red blood cells in a hypertonic solution, the water flows out and the cells shrivel up. In the hypotonic solution, water flows into the cell. The solute concentration inside is high. Water flows in, and cells can actually burst. On the right, we have a comparison of animal and plant cells. And again, a hypertonic solution causes plasmolysis, and a plant will wilt. In hypotonic surroundings, you have the opposite, and the cell can burst. A plant can be hyperturgid. And I think I have, yeah, just to illustrate, and I put this in to emphasize the plant vacuole. Remember, one of the differences between a plant cell and an animal cell, the plants have a big vacuole, and that's for water, primarily for water storage. So in a hypotonic environment, water rushes into the cell and swells the vacuole up and makes the cell very turgid. Whereas under hypertonic conditions, water flows out, and what you get is this, the cytoplasm is all shrunken up, but the cell wall is not going to collapse because it's very rigid, and you just end up with this tiny little vacuole with a shriveled up cytoplasm. Okay, movement of small molecules.
Professor?
Yeah.
Patrick Dowell here. Is that kind of like when a plant's wilted? That's what's happening?
Yeah, that's exactly right. You see the wilted plant here on the left, and it's because, well, it hasn't been watered, and so the solute concentration is very low, and water flows out of the cell, causing the membrane to shrink, and the cell shrivels up within the cell wall. So that's the point I was trying to make here, is that the vacuole makes the big difference in a plant cell. And when it's plasmolized in a hypertonic condition, everything is there, but most of the water is gone and it shrivels. And this is what happens in a wilted plant.
Thank you.
Okay, first let's consider the different types of transport: diffusion, passive transport, and active transport. You can see they're illustrated here in this picture. Diffusion requires no energy and no channel. Molecules can just pass through the membrane from an area of high concentration to low. Passive transport is the same thing, except the molecules go through a channel in the membrane. But again, requires no energy, just passive movement from an area of high concentration to low. Active transport is the opposite. The movement is from low concentration to a higher concentration. So it's against a concentration gradient. It's having more molecules outside than inside is called a gradient. To make a molecule move against a concentration gradient requires energy, requires ATP. Okay, passive transport means it requires no energy. And it can either, as I said, it can either just diffuse through the membrane or go through open channels. And these channels can be either gated or open. An open channel is just a tube to the membrane. And you can see if you look at the bright green dots, they're going from high concentration on the outside to lower concentration inside. A gated channel does the same thing with the pale green symbols. They go through these, what's called a gated channel. It's just not an open tube. It opens on one side and then opens on the other side. But again, this is passive, and you're going from high concentration to low. Now, active transport, on the other hand, requires energy. A transport protein actively pumps a specific solute against that solute's concentration gradient. Systems like this are common in biology, coupling the passage of two solutes. They usually transport both ions as well as uncharged molecules, and always energy is required. That is usually ATP. So what are the functions of active transport? It means that cells can take up molecules against a concentration. They can also move molecules out of the cytoplasm against the concentration gradient. This allows cells to maintain an optimal concentration of ions, also known as homeostasis. Now, the classic example of active transport is the sodium-potassium pump. So this is a channel, a single protein, through the membrane. It's a permanent part of the membrane. It transports sodium ions out from the cytoplasm to the extracellular space by opening on one side and then expending ATP to open on the opposite side, expelling the sodium ions. And again, you see that the concentration outside the cell is higher than inside, and yet the movement is from inside to outside. So it's from a low concentration to high. Similarly, coming back in, potassium ions can enter the same channel when it's open to the outside. And when it opens to the inside, again with the expenditure of ATP, potassium ions are transported in against the concentration gradient. So is that clear? A sodium-potassium pump is a single protein that transports sodium ions out, potassium ions in, expending energy each time. Now, in most cases, it's actually an electrochemical gradient, which means that it's both a chemical gradient and an electrical gradient. And a simple example of that is shown here. with sodium and chloride. Well, sodium is at a high concentration on one side of the membrane and at a lower concentration on the other side. So that's a concentration gradient from high to low. And that causes an electrical gradient so that the charge on the membrane is more positive on one side and more negative on the other. So that's an electrical gradient. And both of these are occurring simultaneously as a result of the difference in concentration of sodium. So remember that an electrochemical gradient has two components. Now remember CFTR, cystic fibrosis transmembrane regulator, is a gated channel protein that requires ATP. And this is just to remind you what we've been over before, that when the CFTR protein arrives in the membrane, it has a certain configuration, which makes a channel. And that channel transports chloride ions from inside the cell to outside the cell. And this needs to happen in order to prevent mucus from accumulating outside the cell. And this is all done with the hydrolysis of adenosine from ATP to ADP. The hydrolysis of this high energy phosphate bond provides the energy needed to transport the chloride ions through the membrane. Okay, so we've been talking about the transport of small things. Let's talk about larger things, bulk transport. And this again requires active transport. There are two types, as you see in the picture, endocytosis and exocytosis. Endocytosis takes large molecules or large objects in. Exocytosis expels them, evicts them, sends them out. So what happens in endocytosis, the membrane invaginates, pinches off into a vesicle, which contains the objects that are being taken up. And then these can be transferred to elsewhere in the cell, wherever they're needed. And when something is not needed, for example, a protein that has outlived its usefulness, can be packaged in a vesicle. That vesicle can fuse with the plasma membrane and expel the contents outward. That's exocytosis. Here's an electron micrograph of exocytosis in action, a vesicle that's fused with the membrane and is expelling the contents. And here you see a picture of it. Protein that needs to be expelled in a vesicle. The vesicle fuses to the membrane, out goes the protein. Now, endocytosis, there are three types. Pinocytosis, which takes up fluid, liquid. Receptor-mediated endocytosis, and phagocytosis. So pinocytosis shown here, taking in liquid. Receptor-mediated, you should immediately think of coronavirus. Think of the spike protein and the ACE2 receptor. That's the receptor that allows the virus to bind to the surface of the plasma membrane. The membrane invaginates, a vesicle is formed, and the virus has found its way inside the cell. Now, for very large objects like a bacterial cell or even larger things, the same process is at work. It's simply that what's being taken in is bigger, and it's called phagocytosis. So a bacterium, for example, when it attaches to the membrane, The membrane invaginates, makes a vesicle, and the bacterium is inside the cell.
Professor, could you back up one slide? And I've got a question.
Go back a slide
Yeah,
here.
yes. So right here where it's showing on the right, the molecules being expelled. Once they're expelled, since they've outlived their usefulness, do they just kind of evaporate, or do they still maintain a function outside of the plasma membrane?
The latter, no, they don't have a metabolic function outside the membrane. They're broken down into amino acids, which then could be retaken up by the cell and reused.
Got it. Thank you.
Okay, that's the end of that slide set. What I've added at the end here is a few summary points to help you think about membrane structure and membrane function. Always keep those two things in mind, structure and function. And if you want to read about this in the textbook, it's pages 63 to 89. Okay, that's it for today, unless there are more questions or comments. If not,
No, thank you.
okay,
Thank you.
thank you. Stop sharing.
Have a nice